Okay, here's the third video. Just like the second video, this one's going to be very short because it's going to be almost impossible to talk about these two processes and make you understand them on this video. Most learning will be done in class. So this is section 3. This is going to be about the central dogma and more likely the protein synthesis. So a lot of times they use the term, people who talk about DNA in textbooks and websites, they talk about the term central dogma. So I thought I would define dogma for you because I'm quite sure it's not part of your vocabulary. It's not hardly ever part of my vocabulary either. So you can read the definition of dogma. That's not going to be testable. That's why it's in black. Remember, you're just going to write down the green stuff. Okay? But basically the central dogma in DNA is, how does our DNA, which contains the recipe to build a protein, get that recipe to the proper place, and then that recipe be converted into that protein? So basically, how does your gene become a protein? So that's basically a two-step process. First step is called transcription. It occurs in the nucleus, and transcription basically turns a piece of DNA into a piece of messenger RNA. Now, just a little note about RNA. RNA is similar to DNA in that it's a nucleic acid. It contains nucleotides. It contains a sugar, a phosphate, and the base. The sugar is different than DNA sugar. And then 3 of the bases are the same. C, G, and A are the same as in DNA, but the base for the thymine base is not present in RNA. So instead there's a base called uracil. So transcription turns a piece of DNA into a piece of messenger RNA. Remember in the previous video I said something about DNA was basically too big to leave the nucleus? Well, that's because it's double-stranded. Remember the double helix? Well, RNA is single-stranded, and it can pass through the nuclear pores in the envelope into and out of the cell. Well, mostly out of the cell. Sorry, out of the nucleus. So transcription makes a messenger. That messenger brings the message to the cytoplasm. In that case, it finds the ribosome, or a ribosome. In there, that messenger is translated into a sequence of amino acids. Now, if you remember, amino acids are the building blocks of proteins. So when we talk about going from DNA to messenger RNA, from messenger RNA into amino acids, we're basically going from DNA to a protein. So one part occurs in the nucleus, one part occurs in the cytoplasm. So here's a static picture of transcription. And notice how it only uses 1/2 of the DNA. So the DNA's double helix is here. It's unwound temporarily. And this big old molecule, this big green enzyme, comes in and takes only one strand of the blue DNA. It's the blue thing, not the red thing. Only one strand. This strand up here is not used. They call this the template strand. A template is like a pattern. If your granny makes a quilt, she might use a pattern for her quilt pieces. So when you pull this apart, this A, T, C, G, A, G, A, T, T, etc., instead of it base pairing up with DNA nucleotides, it base pairs up with RNA nucleotides. So this big green enzyme places those RNA nucleotides, and it generates a single-stranded piece of messenger RNA, which is basically a transcript of the DNA. So it's like a copied version of the DNA. Okay? Once this transcript releases, this DNA goes back together and just hangs out until it's needed again. So that transcript leaves the nucleus. So here's the nucleus, here's that transcript or that messenger RNA leaving the nucleus. And this is where it gets really complex. I'm not even going to try to explain this on the video. But basically, this messenger RNA is read by the ribosome and turns it into amino acids like these little balls you see here, this green, blue, and red balls. So we'll do this in class together. We'll have plenty of practice in this class if we ever get back into class. That's it. Thanks.